Genetic resources can really help us to overcome resistance to pests and diseases. So fundamental within plant genetic resources is crop diversity. We rely on crop diversity, of course, to feed our families. And the world is changing. We know that there's mass migrations going on. We know that we're going to face climate change and its impacts are going to be profound on agriculture. We know that the human race, the global population, is increasing. And FAO has estimated that it's going to take about 70% more production of food by 2050 to feed the 10 billion people we're predicted to have in the world. This is very significant and concerning for most agriculture research scientists. But crop diversity really is fundamental for the entire cycle. It feeds into, of course, conservation, both in-farm and ex situ, or gene banks. And it also provides the resources that breeders need to produce new varieties through pre-breeding, breeding, or participatory plant breeding. And that has implications to farmers in terms of seeds flowing through formal and informal seed sectors. And that is the variety release and marketing that we rely on to get products out and have farmers have money for their livelihoods. So it's a whole cycle, and it's all dependent on crop diversity. We do know that a lot of farming today is relying on monoculture. Monoculture farming is a way that farmers can produce mass amount of foods using mechanization. And it's creating genetic erosion among our plants and our agricultural system. And I think this slide really represents it well, where you can see this large portion of vascular plants that are out there in this world, almost 400,000 of them. And then if you go down to this small piece, you've got about 1/10 of that, or 30,000, that are actually edible, that we can eat and sustain our bodies. 6,000 of those are actually produced agriculturally. And then if you come down here, you have 168 that are actually produced globally, that have any global significance. And then most worrying is the very tip of this, we have nine that provide 66% of the global food production and have about 53% of our daily calories. And those 9 are sugarcane, maize, rice, wheat, potatoes, soybean, oil palm, sugar beet, and cassava. No olive in there, but I'm sure it's up here in the 168 crops due to its importance. So how is the Mediterranean doing in terms of its agrobiodiversity? I wanted to show you this little bit of work. This is actually not done by FAO. It was done by the CGIAR, the Alliance Biodiversity, and SIAT. And what I thought is that it was really relevant to this talk, so I included it. So a couple years ago, they looked at this where they studied the Mediterranean and took 10 different countries in this region and calculated an agrobiodiversity index using about 15 different indicators. And the good news is the Mediterranean world seems to be more agrobiodiverse than the global average. The Mediterranean region is doing particularly well in terms of conservation in gene banks, botanical gardens, protected areas, and so on. Most of the countries were looked at, they had high or very high levels of agrobiodiversity in the market, so people had a good variation for their diets. What was a little bit more concerning is that the production systems in the Mediterranean were less than the global average. So this slide shows some of that data. You can see here the solid black line is the global average. The dotted is the Mediterranean. Spain is shown here in this kind of orangey color. And so you can see in terms of consumption, most of the countries were very good and over the averages. Conservation, also the same, but production, especially in areas like Libya, Algeria, Syria, were much below. So there was calls from action from this study, in that there is a real risk of production systems being dominated, very few crops. You put systems and livelihoods at higher risk for diseases, soil degradation that we heard about this morning, and poor yields. Overall, the Mediterranean had high agrobiodiversity scores, but low production scores. And so there was a call to action to diversify. Also, one request for all countries was to consider monitoring global agrobiodiversity, because as we know, over time things change, farmers change, land use changes, and all of these things can be quite different in a decade. So now I'm going to switch over to talk about some of the policy frameworks of FAO. I somewhat hate to give this right after lunch when we're all stuffed, because it can be sometimes a lot of terminology. But a lot of what we do is we provide technical and policy assistance for the members, the countries that are members to the FAO. And there's about 190 plus countries, so mostly the entire world, with a few exceptions. This slide is very busy. I apologize, I usually don't like to have such busy slides, but I wanted to go over a little bit of the timeline here. And so in 1983, the Commission on Plant Genetic Resources for Food and Agriculture was initiated. And this was a really important milestone because it represents a time where the world came together and said, yes, it's important to preserve genetic resources, to make them available, to share them, to utilize them. And that really came about with the formation of the Commission at FAO. Their mandate has changed since 1983. They used to be plant only, and now they've actually been expanded to all aspects of biodiversity, so it's quite a big undertaking. In 1996, we had the first report on the state of the world's plant genetic resources. So you can think of this as like a big global assessment. So it went out and asked all the countries all these different questions about what genetic resources do you have, what are you maintaining on farm, what are you maintaining in the gene banks, what are your capacities. So the state of the world I'll be talking about a few different times, and just wanted to give you a background of what that means. So we're not just talking about olive here, we're talking about all crops and all plants that you can think of. And then from that, there was a response, the Global Plan of Action, and this is a document that's created by FAO in consultation with all countries around the world. They input on this, and it's basically a guide to help countries know how to institute policies to preserve genetic resources, what they should be doing, different actions they should take, capacity building that they should be doing to secure genetic resources. In 2004, we had the International Plant Treaty that came in force. So for those of you that are not very familiar with that, that was a treaty that basically looked at 64 crops of importance and regulated their access and their use and benefits being shared from them. And so for those of you that work on things that are not part of the international treaty, you're probably very familiar with the Nagoya Protocol, which is a little bit more tricky to navigate. In 2010 and 11, we had a new state of the world, so an update from 1996 on where is the world in terms of genetic resources, and the follow-up, the call for action on what countries should do to secure genetic resources. In 2015, 190 countries signed on to the Sustainable Development Goals, And of course, FAO works a lot on trying to address those. And in our particular area, we work on SDG 2, which is the zero hunger. And that one has been very near and dear to my heart and driven a lot of my work, where I've worked in many different crops and different gene banks throughout my career. Coming up to the end, I'm going to skip some of the stuff in the middle. 2025, FAO just released a new State of the World report on plant genetic resources in March, and so that updates where we are in terms of genetic resources and their conservation. So now turning to the Global Plan of Action, we're currently working on this. We had regional consultations with every part of the world to input on this. And what this document is a document that all countries sign on to and are supposed to implement. And it provides a set of 18 priority areas to enhance efficiency of plant genetic resources and improve their utilization. There's 4 main groups so these 18 priority areas are lumped into 4 groups, and that is in situ conservation, ex situ conservation or gene banks, sustainable use, and one that I find really important is building sustainable institutional and human capacities. I've led several gene banks in my past, and I remember funding cycles going like this, and having stability in terms of your staffing, of your funding, of your mandate is extremely important to keep things going and making sure that you're not losing material that you're trying to save. The Global Plan of Action provides a framework to help countries support policies and institute implementation. And it also is facilitating the implementation of the Plant Treaty, as well as the Convention of Biological Diversity. And this document is so important that the Plant Treaty actually mentions it in Article 14 of the treaty document. So hopefully that's it's a lot of policy information, but I thought that was important and gives you kind of a picture of what we're doing. But we also, of course, deal with technical issues. And again, we are seeds and genetic resources. So we respond to countries' requests for help. And they come to us with all kinds of different requests, anything from in situ conservation, of improving technical capacities, of managing wild and cultivated diversity, to improving knowledge management, to strengthening policies, to how to use technology. We also work in collaboration and respond to requests from gene banks. And their mandate is to collect, characterize, evaluate, distribute, and safely duplicate germplasm. And we also, the gene banks also do a lot of documentation of information. They do biotechnology, as well as a lot of large-scale phenotyping, and we help them overcome technical hurdles in these areas. We also are very focused on sustainable use of genetic resources. It's one thing to preserve all of these plants and crops for our future, but if they just sit there like they're in a museum, that does anybody any good. And so we want people to really use these resources. We want engagement. And so some of the areas we respond to is strengthening plant breeding, which is, of course, really important for the development of new varieties in our agricultural future. We also have a team of people working on strengthening seed systems. So in adoption of new crop varieties, seed production, seed testing, how do we know that the seeds going out to the farmers are actually what we think they are, and they aren't loaded with disease that's going to just wipe out, entire crops. We do capacity development of testing labs and quality assurance, as well as emergency intervention. We all know disasters occur. Of course, nobody wants to see disasters occur, But when things like a hurricane or a typhoon comes through, like the recent one that happened in Asia, farmers lose everything. And so we help get them access to quality seeds and planting materials, as well as supporting local production of quality seed. We also develop a lot of guides. And so these guides are to help countries and members of FAO deal with various bottlenecks they have, anything from national seed policy to quality declared seeds to seed toolkits. We've developed guidelines for national strategies for plant genetic resources for food and agriculture, voluntary guidelines for conservation and sustainable use of crop wild relatives and wild food plants. And you might notice that they get translated in all the languages of FAO so that they're more useful, right? Because if we only publish in English or only publish in Spanish, we limit who can actually utilize this type of information. We also have created gene bank standards. These are very thorough standards that all gene banks are supposed to strive towards, to have a cutting edge facility. And we've developed practical guides. So how do gene banks deal with things like non-orthodox seeds? Seeds that don't dry down and don't store in the freezers very well. How do you deal with clonal crops? How do you deal with in vitro cultivation or cryopreservation? So we develop the guidelines and help countries overcome these technical hurdles that they may have. So this is just a kind of basic overview of our work cycle. As I talked about, we have the assessment where all countries report where are things in terms of genetic resources, where they're at, what gaps do they have. And through that, there's a response, a policy response in the global plan of action. And then from there, we notice that countries have certain bottlenecks or certain issues, so we try to address those by developing various guides to help everybody out there. And then we monitor what's going on, and then the cycle repeats. And so it just kind of gives us an update of every 5 to 10 years, where are things in the world, what needs addressing. And we use this website called Views, some of you may have heard of it, to help monitor and house a lot of this data. So for any of you that are very interested in this, here is the QR codes for the State of the World's Plant Genetic Resources. You can scan these and get the full report or the in-brief downloaded, and also in the FAA website, or by searching it, you can easily find it as well. So this slide just shows some of the data output from the Global Plan of Action, or the implementation of it, or where countries are. You can see the ones in dark green here reported on the Global Plan of Action, so what the status was, where they're at. And they also reported on SDG Indicator 2.51A. So SDG 2 is zero hunger, 2.51A is a measure of what genetic resources are out there, how many are in different collections that are stored in medium and long-term storage. So for example, what olive collections are out there and how are they stored. And so some countries reported on one, but not the other, some on both, and a few countries we have no information available. Reporting on this, I can tell you, is quite intensive. I was on the other side of this, having to report for the Global Plan of Action, and it is a lot of work, not only for FAO, but for the countries. And so we're in the process of trying to simplify this. So Views, as I've mentioned, is a website that you can easily find in Google. It's a world information and early warning system on plant genetic resources. Currently, there's records on 6 million different plants in this site. But we use it to monitor the data from countries on the Global Plan of Action, as well as the SDG 2.51, which is what is in collections. So now turning to olive, I've talked a lot about FAO, but we're here for olive. So is olive important to protect and conserve? And you know, to me, the olive is so important to the fabric of Mediterranean culture and goes back so long in its cultivation. And just, you know, quick search online, I just couldn't believe how many references and symbolism I found to olive. Even the United Nations flag, where the world is wrapped with the olive branch, you know, symbolizing peace and harmony. Or how many religions use olives, you know, to symbolize faith and harmony and peace. And of course, this Vincent van Gogh painting of olives. And I really like this. I didn't know that the goddess Athens was thought to have put her sword in the ground and an olive tree grew. And of course, this last one is one of my favorite, that ancient olives from Greece went up to space, came back down, and were planted in Japan to you know, promote harmony between the two countries. So to me, olives are extremely important for conservation, really to preserve the culture and the richness that they have provided to this region and beyond. So a snapshot of what we know is conserved from views. There's a little bit shy of 4,000 olive accessions of 8 different species that were reported by 30 different countries to FAO at the end of 2024. So we get this data on an annual basis at the end of the year. So this is the last year that we have data for, and in 2025 we'll get an update. The majority of these accessions are conserved in Italy and Spain, which makes a lot of sense, especially coming here and seeing all the olive trees. It makes a lot of sense to house them here. The one thing I wanted to note is when you look at some of the relatives of olive, the holdings are very, very low. And so to me, it seems very important to increase those holdings and make sure that those species are not lost, because they could contain genes that provide disease resistance or other traits important to olive in its future. Another way FAO has been helping in terms of olive is in 2013, you probably all know about the outbreak of olive quick decline disease, a really nasty disease that wipes out the trees. And FAO has been supporting preventative efforts, mainly in Algeria, Egypt, Lebanon, Libya, Morocco, and Tunisia. So it did a lot of stakeholder consultations, a lot of training sessions, a lot of diagnostics and surveillance and looking at management practices. FAO has tried to work with different countries to also increase their phytosanitary legislation so that this disease doesn't move into more countries and just spread like wildfire. So FAO has been supporting testing of imported material to make sure new olive material coming into a country does not contain this disease, and using what's known as LAMP testing, which is a great test because it can be done in the field and a farmer doesn't have to wait for weeks for a sample to go to a lab and the results to come back. So we really hope with some of these interventions that olive cultivation and its history can stay as it has been and not turn into something on the right where the trees are just ultimately just decimated. So in wrapping up, challenges in promoting the diversity of PGRFA. A lot of times plants and crops are underrepresented in gene banks. And as you saw from what I showed, a lot of the olive species, according to views, are very much underrepresented. And so a lot of action needs to be taken in terms of preserving this material and making sure that it's available for our future. There's often a lack of knowledge on conservation status, especially on farm or in situ. It's very hard for people to monitor the world and know what every farm has or what's in the wild in every place. So there really needs to be a lot of effort going into that. There's always need for increased technical capacities, infrastructure, and sustainable funding. As mentioned, I worked in gene banks most of my career, and funding went like this, and it was really hard to know year to year what you could and couldn't do in terms of preserving plant genetic resources when your funding was all over the place. Bioinformatics and biotechnology are very well advanced now. There's so much that can be done with it and is being done in plant genetic resources. But often national programs don't have these capacities or don't have the technological resources. And so a lot of capacity building is needed to uplift some national programs. There's typically limited partnerships on stakeholders and insufficient linkages between ex situ and in situ conservation. And I would go one step further. In my experience, there's oftentimes in many crops kind of a divide between the gene bank and the breeders. They're not always talking, they're not always working together, and that's a big opportunity missed. So with that, I'd like to close. I just thought I'd close with this quote because I thought it was quite relevant. Just as the olive yields oil for light only when it's pounded, so are man's greatest potential realized only under the pressure of adversity. And I think when we think about agriculture and farming and our future, we are in a challenging time in terms of land use, of climate change, of new diseases and pests that we're going to have to deal with. And hopefully all this adversity is going to make us stronger and new great things are going to emerge from it, just like olive when it's pounded. Thank you so much for your attention. This is not just my work. This is a lot of other people involved in the Seeds and Plant Genetic Resources team. I would be happy if you contacted me and I could put you in touch with others if needed, if the question is out of my area. And with that, I will close. Thank you.
Okay, now everything is working. Good evening. Thank you to the organization for inviting me to this amazing place. It's a bad timing because it's after lunch, so I will try to be energetic with the presentation and engaging. So I will tell you the story of a project. Its name is Gene for Olive. It's a project that it was born from our fascination about genetic resources, also from our fear of losing these genetic resources and our will to do something. So we know that olive diversity, olive genetic resources are composed by the cultivated olive with a large legacy of traditional cultivars, the wild olive that is part of the spontaneous vegetation of the Mediterranean basin, and also by ancient olive trees that can be considered in some cases like living fossils. But this rich genetic diversity is under threat because of several reasons. First, intensification. The intensive and super high density plantations are spreading worldwide, and the homogeneity, genetic homogeneity of this plantation is dramatic. So only 2 cultivars, so maybe 5, adapt to these high-density plantations because have the low vigor required to these super high-density numbers. Also, of course, climate change. Climate change is modifying the patterns of rainfall, temperatures, and is leading us to see like disturbing patterns. For example, asynchronous flowering. Asynchronous flowering can be normal here in Canary Island in this amazing picture, but maybe it's much disturbing to see it in Cordoba. In this picture, we have in the same branch set fruits and flowers in May, in the main producing area in Cordoba. Why is this happening? Also, climate change is contributing to the spread of a known disease like verticillundale and new ones like Xylella fastidiosa. All these threats also are affecting the wild olives. Wild olive populations are being fragmented. The introgression from the cultivars is increasing. Also, ancient olives are being pulled out in many areas before we know which cultivar they are. Are these ancient cultivars cataloged? Do we know something about them? So, in this scenario, can we do something? Yes, we can do something. We can preserve in all its forms, ex situ, in situ, on farm, but we realized that the connection between germplasm banks was very poor. Some germplasm banks, and there are many, there are like 23 germplasm banks of olive genetic resources in the world, but they were poorly connected. Some collections were like just collection of accessions of names, and the wild olives were not represented in these collections. So it was necessary to characterize the genetic diversity, identify the cultivars, characterize them agronomically. Also, this action will lead to efficient conservation, but also they are crucial for breeding. Breeding new cultivars adapted to changing environmental conditions is key in these moments. But the breeding in olives is germinal. If we compare it with the breeding in apples, in peaches, it faces also important methodological issues, for example, derived from the long juvenile phase of olives. But breeding, the main barrier for breeding is that we know a little about our traditional olive cultivars. For example, if I ask you, which are the 5 most tolerant cultivars to drought, could you give me a well-supported answer for that? To frost? And that is the main limitation for breeding. We know a little about this huge diversity that the olive have. In this context, we thought that an international approach to evaluate genetic resources, promote efficient conservation, and boost breeding was needed. So, and in this way, in this context, Gene for Olive was born in 2020. It is, or it was, for me it's weird to talk in past tense because the project finished a few months ago. Gene for Olive started in 2020, finished 4-5 months ago, and it gathers 16 partners for 7 countries to pursue 3 main goals. First, characterize germplasm, characterize the genetic diversity of the olive oils. We hope to find the genetic diversity that we expected in this material. Also, to make this information accessible to end users, and also try to promote the collaboration of the private sector, the public sectors in olive breeding. So in this talk, I will focus on the first, goal, the characterization of olive genetic diversity. What we found in this project. Okay, in this task, the main actors were 5 international germplasm banks of olive. Of course, we are talking about olives in the Mediterranean basin. One, Marrakech, Cordoba, Rende in Italy, Crete in Greece, and Izmir in Turkey. Altogether, we committed to evaluate traditional cultivars. So the first step was to elaborate common evaluation protocols. We wanted to measure the same traits using the same protocols, even the same machines. So we elaborated 14 protocols, and with these 14 protocols, we evaluated more than 40 traits in more than 500 cultivars. And what did we find? For example, phenology. Phenology, we found that flowering is a very explosive and ephemeral process in olive. This is an histogram. I will show many of these in this presentation. We have the number of cultivars that flowers in these days. And in the 5 germplasm banks in the whole Mediterranean, olives are flowering within a week. Within a week, imagine a collection of 500 cultivars. Within a week, almost 90% of the cultivars flower. So there is almost no variation in flowering time. So makes sense try to develop a breeding program focused on selecting early flowering or late flowering cultivars. Maybe not. Maybe olive has a little diversity in flowering. But we knew that bud burst is a more steady and variable process. Maybe the adaptation of olive to changing temperatures could be in bud burst, not flowering. Also, draw. Draw is a very, very difficult trait to be evaluated. It's a very complex trait. And we had to evaluate many cultivars in many germplasm banks with different levels of equipment. So we developed very easy to apply protocol. How do we measure draw? We calculate the minimum leaf conductance. Simply is the amount of water that a leaf loses once it's detached from the tree. So these are the results of the bank in Marrakesh. This is in Izmir. And we have here cultivars that lose a lot of water. Then we can consider them that are susceptible to draw. But we have cultivars that lose few water. We can think that maybe they could be tolerant to draw. And you will think, this is a loose method. Okay, it is. But it provides a baseline. Now, if you want to run more detailed analysis, you have something from where to start. Took the tails of this distribution and run detailed analysis. So the same with diseases. This, another histogram. These are the resistance, the incidence of Spilothea and Coletotrichum in the germplasm banks. And again, we have very susceptible cultivars, but we have many that we can consider them resistant. So there is a huge diversity in the behavior of traditional cultivars, for example, regarding aerial diseases. What about oil? Because at the end, these plantations, okay, there is table olives, but we want to produce oil. About the oil content in the fruit. This is the histogram of the cultivars, and we can see here where Arbequina is. Arbequina is a well-known cultivar, but most, it just, okay, thank you. Most of the cultivars show a higher oil content than Arbequina. We went further because we wanted to know about the composition of the olive oil. And this is, okay, this is the composition in the oleic acids. Here we have the three fraction, polyinsaturate, monoinsaturate, and saturate fatty acids, and the different the different acids. You can see that the large diversity in all the fractions. For example, let's see oleic acid here. We have cultivars that are below 55%, cultivars that are, their oils are about 55%. These are the limits to consider an oil pure by the International Oil Council. So according to these results, maybe the IOC should reconsider their purity policies because all of them are extra virgin olive oils. Okay? Also, we study, were these characteristics stable across different environments? Then we also study the genotype per environment interaction. And here we have the three fractions of fatty acids, polyunsaturates, monoinsaturates, fatty acid, and the influence of the cultivar, the environment, the interaction between two. And we knew, this was already reported by smallest size studies, that the composition in olive oil is quite stable, independently of the environment. It doesn't happen the same with phenols. Phenols are quite variable. The genotype explains only the variation of a small fraction of the phenols, but not of all the phenols. For example, we have here the methylated forms in which the cultivar is the main source of variation. What about wild olives? We have several partners just focused on wild olives. In my opinion, is the forgotten partner of olives. And maybe, of course, the diversity of the cultivar is huge, but the diversity, we don't know the diversity of the wild olives. So the main part, one of the main parts was to identify where these populations were and to collect them, to sample them, and study their genetic diversity. We did that in the whole Mediterranean, not only in the oleaster, the ancestor of the cultivated olive, but also other subspecies. For example, it's very interesting, Olea europaea subspecies Laperini. Laperini is in the Sahara Mountains, and we can think that it can be very resistant to drought. But we don't know anything about this subspecies. We have sample population, only few populations exist of this subspecies, and we have to evaluate it. We found wild olives growing in very hard conditions. For example, these are marshlands here in Spain. This is the Ría del Odiel in Huelva. This is salty water, and we have wild olives growing in these conditions. Also, we have nice surprises regarding the resistance to diseases. This is, again, an histogram, and here we have the disease severity of Coletotrichum. Okay. This is the distribution of the wild olives that we analyze, and here we have the cultivar picual that was used as control and is considered resistant, and this is the cultivar frantoio that is considered highly resistant, another control. So we can say that most of the wild olives were resistant to Coletotrichum. So wild olives can be a priceless source for disease resistance and adaptation to different environments for the cultivated olive. There is not a single collection of wild olive seeds in the world. So the population that we have, if they disappear because of wildfires, fragmentation, they are lost. There is not a single collection of seeds of olives. So in this project, we created the same collections, and we want to send a duplicate to the Svalbard Island in Norway. I don't know if you know this facility. It's a gene bank in the Arctic where the seeds are stored in case of a big disaster. We want to create the collection, but also send a copy here to safeguard this genetic diversity. And you can say, okay, where is all this information? Where I can find them? They evaluated 500 cultivars. Where is this information? This was the second goal of the Gene for Olive. And we built a database that is available now, where we store all this information. You have information of more than 600 cultivars. Here in this QR, you can access the web page. It's open, it's free, you can use it, please use it. Please share this link. Here you have, you can check or look for a specific cultivar, and then you have the pictures for a phenotypical characterization, and also all the results that we got in about disease resistant, pest resistant, also agronomical traits, load, force retention fruit, etc. So this is the main result of Gene for Olive. So please use this database, share it, because I think that is something that has not been done in any other fruit crop. There is nothing similar in peaches or in apples. I think that it's beautiful that just for first time, maybe, olive is ahead apples and peaches in something related with genetic resource conservation and science. So the main result of Gene for Olive, apart from this database, is the diversity. The diversity that we found in the cultivated and the wild olive. We have these histograms. What we see here is diversity. We have cultivars that are very resistant to frost. Here, very susceptible. Here, in oil content, maybe flowering is not the best example, but this may be the only one. The big result and the beautiful result is that olive has a huge genetic diversity. And this genetic diversity is the key for olive resilience and guarantee the long term of this crop. So if you are involved in any of these, if you can preserve, if you can identify, if you can use the information for breeding, please take action. Take this material, preserve this material, identify this material. So because it's a responsibility of all of us to preserve the genetic diversity of the olive. So thank you very much to all of you. Thank you.
We thank you, Concha. I mean, I think it's easy to recognize how important this work is. Congratulations for accomplishing. Time for a question, maybe, yes? Any questions? Please, Antonio.
Thank you, Conchi, for this nice talk. So I know the project, the Gene for Olive project, but I'm also very interested in natural variation of complex traits and other traits that you normally adapted traits sometimes are complex, especially some that are quantitative. So I'm very surprised about there is no correlation between flowering time and brotation. So there are not correlated, at least phenotypically, it should be a correspondence between them, but it seems that there is no genetic correlation. I mean, you say that there is no much genetic variation in flowering time, but yes, in the other traits. I
mean, this is a very good question, but I mean, I didn't address this here. I just talk about the diversity of flowering, the diversity of bud bursts. Are they correlated? Yes. Barely correlated. The correlation is low. Also because bud burst happens during a month and a half, flowering happens in 1 week, 10 days. There is correlation, yes. But what you consider early bud burst happens 1 month before than what you consider late bud burst. What you consider early flowering is 7 days before that what you consider late flowering. So there is a correlation, yes, but it's very small. Also because the flowering is so explosive that you have almost no variation.
So this is my second part of the question, because to me, with my experience with olive, is that probably one of the main challenges for the future of the production of the olive oil is the temperature we are going to have in the Mediterranean during the spring. Not summer, spring. I'm talking about March, April, with heat waves that literally will destroy flowering. It happened. So my question is, what is the best strategy from the breeding point of view, or to try to get new cultivars to overcome this problem that we are going to have now?
I mean, this is, and I am passionate about this topic, so I will try to be brief because I could be discussing about this very long. The temperatures during the spring are dangerous because they can burn the flowers. But in my opinion, what is really dangerous is that we know that for flower induction, we need chilling. What is chilling in olive? That is another question. But we need a certain amount of chilling. And what happens if the winters are not cold anymore or not enough cold? We will have no flower induction. That is what happened in the Canary Islands, in Peru, in Mexico. Is the olive not growing? It's growing amazingly well. It's a very huge tree that vegetates perfectly, but never flowers. Why? Because there is no chill. In the moment you have chill, you have flower induction. So temperatures during winter, not having chilling is very dangerous. What happened between bud burst and flowering? The hotter it is, the sooner the flowering. So if the flower is sooner, maybe it can escape from very hot temperatures in May, maybe. But the problem now, for me, the most problematic about climate change is not that temperature increase on average 1.5 degrees. For me, the most problematic is that we have erratic temperatures. And we cannot prevent something that is random, that is erratic. Because if, for example, in January, we have days with temperatures about 30 degrees, that erase the chilling that has been accumulated. And that is quite dangerous. What happens also if in spring we have chilling conditions? And that happened here. We have chilling 12th of April. I don't remember exactly which year, 2016. That one, the one that, okay. The one in which I show the asynchronous flowering. What happens if during spring, with new buds, I have chilling conditions? Chilling induces the buds that had been formed a month ago. And then we have that. We have set fruits and a wave of flowering. So, yeah, temperatures is not only the temperatures in April. In my opinion, the most dangerous of climate change is the erratic temperatures. You cannot prevent something that is random.
Thank you, thank you.
Yeah.
Do we have room for another question? Rey Zamora from the University of Jaén. We're going to talk about demonstrative projects of how we can enhance all these aspects we're talking about, resilience. Yes, so the floor is yours.
Okay, yeah, it's working. Thank you to the organizers and scientific committee for inviting me to give this talk. And like Basileos tell us, I am an ecologist, so now we have to move the target from genetic resource for food production to the nature, or at least to the wildlife biodiversity. So the aim of my talk is to show how agroecological theory may be used to inform restoration action to recover biodiversity in permanent cropland. I am remark this issue of permanent cropland because it's time to pay attention to permanent cropland as the most cropland, or the cropland with most potential for retaining, for hosting biodiversity, at least compared to annual crops, for many reasons. I am not to read the reason, but they have been extensively discussed in this article recently published in Nature Sustainability from one of the PhD of my lab. I prefer to talk about why olive orchard. Why should also pay particular attention to wild olive orchard in order to host biodiversity. And there are many reasons. Many of them are similar to the previous one, like olive orchards are the most important permanent cropland in Europe and among the 3 most extended in the world. Overlaps its cultivation area in the Mediterranean region practically totally with the biodiversity hotspot of the Mediterranean basin, which is the most important biodiversity hotspot in Europe, has potential for biodiversity refuge for Mediterranean flora and fauna, which is linked to the previous reason, forms part of the cultural heritage of the Mediterranean, as has been already commented here, and because olive yield sustainability, biodiversity, and culture are threatened by agricultural intensification and climate change. So, which are the main dimensions of farming intensification in permanent cropland that are impacting on biodiversity, and how we can, or what effect they are having in this biodiversity. In my view, there are three dimensions of intensification in olive orchard. The first one is a ground management and agrochemical intensification gradient, which has supposed transforming olive groves from living persistent ground cover, which the many advantages that has been already commented in this conference, to olive orchard, olive groves that maintain bare soil most part of the year, with all the environmental concerns that this has. There is another important gradient of intensification, which is probably less acknowledged, which is the transformation that agricultural landscape is suffering from landscape with high representation of semi-natural habitat cover to monoculture of permanent cropland, or in the particular case of olive orchard, monoculture of olive orchards. And currently we have, or more recently, we have another important gradient of intensification affecting biodiversity, which is the transformation of traditional frames of permanent cropland, in the case of olive trees, from less than 100 of olive trees per hectare, a hedge-like plantation, super intensive hedge-like plantation of thousands of olive trees. And all of this has parallel, or has influence, or has considerable impact on many groups of organisms, both from flora and fauna. And there are a lot of evidence supporting this decline. So, my aim with this talk is to show how modern agroecological theory can be exported to active restoration project, how inform about how conducting restoration project, with the central idea that I defend that the heterogeneity of the agricultural landscape sets the arena for agricultural intensification to impact on biodiversity, conditioning the level of loss and the possibility of recovery after ecological intensification. So what does agroecological theory tell us about the impact of agriculture intensification on biodiversity, and what environmental factors moderate this impact? I have no time to explain the conceptual idea, but I would remark that there are three main theories around the impact of agricultural intensity in biodiversity. The first one is the land use intensification hypothesis that do not pay attention to the landscape context. The second one is the ecological contrast hypothesis that pay only marginal attention to landscape context. And the third one is the landscape moderation of biodiversity and ecosystem, ecosystem function framework that pay all the attention to the landscape complexity or landscape context. So let me go in depth in this last framework, which is not only one hypothesis. There are many hypotheses associated to this framework. And basically, I would remark two of the main hypotheses of this framework. That is the intermediate landscape complexity hypothesis, represented by this two figures, in virtue of which, based in theoretical ecology, there are an increase, a nonlinear increase in biodiversity and ecosystem service with the increase of landscape complexity. And this nonlinear increase means that they are some point of saturation, so at intermediate to high level of landscapes saturate the biodiversity that a landscape can host. Okay? And this trend is similar to some extent in very intensive agriculture or in low intensity agriculture. But when but there are differences. Always we have the biodiversity in intensive agriculture below the biodiversity of intensive agriculture. However, in virtue of this nonlinear relationship, it comes that the difference between intensive and extensive agriculture are relatively small in simple landscapes, in very simple landscapes, and also in very complex landscapes. I have no time to explain why this model proposed this, but I can answer a question, finalize the talk. So the differences are fundamentally happening at intermediate level of landscape. So when we move through applying agri-environmental scheme, through ecological restoration, or through applying wildlife-friendly agriculture, like organic agriculture, the recovery of biodiversity, the effectiveness to recover biodiversity and ecosystem service will peak at intermediate level of the landscape. Okay? This is important because recovery of biodiversity, effort to recover biodiversity may be not practical if we maintain determined level of landscape complexity. The second hypothesis is within the framework is the hypothesis called the dominance of beta diversity in the landscapes, that basically state that when we modify landscape by agriculture, transforming heterogeneous landscape to very simple monoculture landscape, we are losing habitat, and parallel with this is happening a loss of biodiversity at the landscape scale, which is called gamma diversity in ecology. But this loss of biodiversity is happening fundamentally by the component of the gamma diversity that we call beta diversity, which is the difference between habitats in diversity, okay, in the biodiversity they host, but not too much in alpha diversity, which is the diversity within habitat. And this remark also that if we want to retain biodiversity or to recover biodiversity, we need heterogeneity in the landscape. We need habitat heterogeneity in the agriculture landscape. These are the main conceptual ideas, but how can we extend these ideas to create an experimental demonstration framework that we are able to recover biodiversity using this type of information? Right? So we need experimental setting. But this experimental setting for recovered biodiversity needs to be conducted through landscape gradient and through intensification, agriculture intensification gradient. Okay? And we need, at this position of this gradient, operate not only with experimental plot, but also with control and reference system in a 3 stages recovery project or restoration project. Of course, in any restoration project, we need an intervention stage. In the case of agriculture, we can increase landscape heterogeneity, we can change from intensive to extensive agricultural practices, or we can incorporate a small infrastructure to support fauna. Okay? But we need previously to have diagnostic or diagnosis of the stage of the biodiversity, of the stage of the landscape, or the state of social and economic profitability, previous intervention. Okay? And we need also to measure same indicators several years after perturbation, after intervention. And it will produce, as a result of comparing post-intervention with previous intervention, change in biotic and socioeconomical indicator associated with the restoration. This could seem enough, but not is. Why? Because, unfortunately, this will produce recovery index that are not stochasticity free. That is, that are influenced by the demographic and environmental stochasticity, by the environmental nature, by the stochasticity nature of the biodiversity. So, to control for this stochasticity, we need also operate with control, in which we are going to measure same indicator of biodiversity or same indicator of social and economic profitability, same indicator that we are using in the experimental plot, and we should do both. Should do it both in pre-operational and post-operational state. Also, these controls are not in intervention. And this will allow to control for this stochasticity and thus produce stochasticity-free biodiversity and ecosystem function recovery indexes. And this, again, could seem enough. But if we want to know not only if we are recovering biodiversity, but also which kind of biodiversity we are recovering, at what rate, and how far we are of the optimum biodiversity of the territory, we need also use appropriate reference system that would indicate the appropriate biodiversity of such land. So this is what we call before-after control impact design in studies of ecology. And a design like this can produce recovery indices truly free from stochasticity, as the difference between demonstrative and control fund of each locality of the chain each experiences in biodiversity between the pre and post-operational stages, which can be, which respond to this equation, which can be standardized to be comparable index between different indicators of biodiversity. For example, standardizing it as a proportion of the pre-operational biodiversity of the demonstrative fund, which, I said, make this truly comparable. So, this is the conceptual idea. This is the experimental framework that we can use to demonstrate that we are recovering biodiversity as a consequence of our intervention. So, let's pass to the action. And I will suggest that there are some umbrella funding programs that allow now to work agroecological research, active restoration, and olive farming together to perform this restoration project. So I am going to present, to introduce one project in which I was working for a decade, that is the long-term biodiversity recovery demonstration project Olivares Vivos, that is based in this conception. Olivares Vivos is a project LIFE, it's a LIFE project, assessing to what extent the change provoked by biodiversity-friendly agriculture and the implementation of restoration plan renders certain enhancement of biodiversity in olive orchards from Andalusia. We are developing an eco-scheme that has 3 main pillars. One pillar is sustainable management of the herbaceous cover that aims to maintain hair ground cover most part of the year in the olive orchards. We have a second pillar that is forest restoration of not productive areas that look at or that intend to generate heterogeneity, and we are installing some small infrastructure to help fauna, to support fauna. And this is conducted as a before-after control impact experiment, like I presented before, across a gradient of landscape complexity in the most important area of Andalusia, and across a gradient of agriculture intensification. This project was first initiated in 2016 in 20 demonstrative olive farms in Andalusia, and it is now extended and replicated in a second LIFE project to other 3 regions in Spain and to several regions in Portugal, Italy, and Greece. And we are using many different organisms as indicator of biodiversity. We are considering many groups of flora and many groups of fauna, and within fauna we are considering both vertebrate and invertebrate indicator, and we are measuring all of these previous and after intervention. And there are some of the more important results. We have a lot of results that I have not time here to present, but this project has some evidence supporting that olive landscapes still harbor a notable amount of the biodiversity of the Mediterranean region. In fact, on average, 1/3 of the total biodiversity of flora and fauna of the Iberian Peninsula is still present in the olive landscapes. Okay? So this allows me to send the message that olive orchards are strategic for the conservation of the European biodiversity. Okay, we have also confirmed some of the postulates of this agroecological theory that I presented. That is, that biodiversity in these olive orchards largely varies across site and group of organisms due to variable level of landscape simplification and intensification of agriculture. With the optimum, we have identified which is the optimum for overall biodiversity. This could be a good reference system for the biodiversity that is possible to host in olive landscape. That is organic farming practices, living herbaceous ground cover within complex landscape. And this has similar results for many different groups of organisms. Here, green colored, the organic or less intensive agriculture. We have also demonstrated that there is important loss of biodiversity at the strength of this intensification gradient that I presented before. For example, in the strength of combination of ground management intensification and landscape simplification, which are those olive orchards that live bare soils in homogeneous monoculture landscape, produce loss of biodiversity of more than 20% of bird species, more than 30% of the ant species, more than 30% of the flora species, and around 50% of total abundance of bees. And something comparable happened at the extreme of the intensification gradient of tree density, with very important loss of flora, fauna, and birds in particular at the hedge-like plantation. We are advancing to inform, for example, to the Common Agricultural Policy about proper result-based agri-environmental measures, because we are identifying threshold of environmental change and critical point for multi-species synchronous response in abundance. Okay, what this means? Means that, for example, using bears, we have detected an unavailability threshold, which is associated with pesticide and soil distribution, and ants are used as normal food by bears, and we have situated a critical point of 85 ants per week per pithal traps that lead to a steep increase in abundance of multiple insectivorous bears. We have detected an herbaceous and woody plant richness threshold in the olive farm with around 85 species of herb and 15 species of woody crops that increase overall the abundance and diversity of birds in the olive orchard. And we have also detected a natural, semi-natural cover of forest in the landscape that enhance the diversity of most of the birds and that need a critical point of 15% cover, okay? But this needs to be even higher for forest specialists and only cause decline in granivorous bears. So, what about the recovery that we are achieving applying the Cosquén olivares vivos? So the Cosquén olivares vivos have rendered a short-term recovery in overall biodiversity and in some specific groups. Recovery was especially notable in intensive farm, and this recovery is happening in richness, but it's especially important in abundance, which is not rare because abundance is normally a parameter that responds quickly compared to species richness. Okay, so we are able to recover biodiversity with an ecosystem like I have presented. But how much diversity can we recover, at what rate, and how far we are from reaching regional biodiversity pool or an optimal of biodiversity as should be our reference system. In order to answer to this, we are using a recent proposal that is conceptual proposal that is called the recovery depth approach. It's very complex, mathematically even more complex, but I will try to explain in a simple way. When an important and maintained perturbance through time is happening, normally it causes an important decline in biodiversity. Okay? And when this disturbance, for example, intensive agriculture ends, and we start to change to biodiversity-friendly agriculture, or when we apply restoration action, start the recovery of the biodiversity. And the start of biodiversity, the recovery of biodiversity progress in time. And this progress can be modeled to approach the reference value of biodiversity in such territory by mathematical approximation. This is what is called the distance to the reference is what is called the recovery depth, and we can model this recovery depth. We have an organic extensive farm in complex landscapes at the reference system validated in olive orchard because this hosts the maximum biodiversity that we can retain in olive orchard. So we apply this to different groups. I am going to here to show the case of pollinator to vary the type of target group. We have confirmed the decline over time, shortening in recovery the depth of abundance, and also in richness. Shortening means decreasing the recovery depth between different intervals, time intervals. Okay? We have also information of what practices, of what intervention measures have accelerated the process of the shortening the recovery depth. And we have information that extensive management of the ground covers and increase in the quality of floral patches is responsible of this shortening of the recovery depth for pollinators. And finally, we know also which state or which circumstances of the olive orchard previous to the intervention favor also the recovery depth shortening or accelerated of the decreasing of the recovery depth, which are landscapes in which there are many cover of semi-natural habitat, non-forested, because pollinators are favored by other type of habitats like prairies. So let me conclude with the main messages of this talk, that are that olive orchards are still one of the most important refuge for biodiversity in the Mediterranean hotspot. However, the amount of biodiversity harbored at each locality is contingent on agricultural intensification and the heterogeneity of the landscape. That is possible to detect threshold and tipping point of environmental change to inform proper result-based agri-environmental measures, which is completely needed to correct application of common agricultural policy. That the short-term biodiversity recovery is feasible by biodiversity-friendly practice and active restoration of non-productive areas, not compromising production. That Olivares vivo for core is an eco-scheme efficient for short-term recovery of biodiversity, and that despite this efficiency, still recovery debt, an important recovery debt will, however, remain. So we need to maintain green and fauna supporting infrastructure and biodiversity-friendly agriculture through time. Okay? So, of course, thank you very much for your attention. Thank you to the research team of Olivares Vivos, which is represented in part here. And thank you very much to all the funding institutions and programs that are funding this project.
Thank you, Pedro. All right, I don't think we have much time for any question. I think it's a very important project, yes. An added value also given for the farmers, which is very important. They can tangible results that can show off. You know, they can show to. It is obvious that how all these biodiversity-based approaches enhance biodiversity. I will give the floor directly to colleague Professor Emmanouil Kabourakis from the Hellenic Mediterranean University from Crete, Greece. He's going to talk about a rather twin sister project, LIFE IGIC. Yes?
We'll go back to agronomy, actually. It was very nice, the introduction of Pedro, because like it's an introduction for me, because it's based on the theoretical framework that already discussed. I will talk about the LIFE IGIC project for green infrastructure. And I'm glad that Vasilis is here. He had been working in this project. So we have the same aim, so to conserve or increase biodiversity in olive orchards, but also as agronomists, for me, it's important to sustain production and to adapt and to offer the maximum of ecosystem services. So I'm not going to details this. This already have been discussed. We need to restore, enhance biodiversity, enhance sustainability, all these things. I think it's already done. And the problems have been discussed. The main problem at the moment related to biodiversity, and this is not discussed, we discussed about the olive tree and the effects of the climate variability to the olive tree and the production, which is the most important, but there is also effects to the biodiversity elements, flora and fauna. And we have little information, we need little about this. Now, about this project, which is called Improvement of Green Infrastructure in Agroecosystem, reconnecting natural areas by counter habitat information, because actually olive production areas usually neighbor to natural areas, like Natura areas. This is a national project actually done in South Crete, in an area that is surrounding by 2 Natura areas, and we have a diverse landscape. Crete, I don't know if you visit this, has already a diverse landscape, so we have what we call 2 agroecological zones. One plain, a flat, and one hilly, which is different landscape and different complexity. So how we work also with different olive farmers. It's a demonstration project, and we choose also organic and conventional. That's because for the same reason, organic is supposed to be more reliable and so on, and more diverse, and we wanted to check this. So we focus on Halting biodiversity practically, because we work in a rural area. So for the farmers, how we can conserve and increase and sustain flora and fauna diversity. What are the practices? Because we already saw the negative effects of wrong practices or unsustainable practices, and how to optimize agroecosystem services. So that's the main aims. And so for this, we designed, like the Olivares Vivos, we designed how should be green infrastructure, how something to support in olive grove the biodiversity. And this relates to the methods, to the practices that the farmers apply. And actually, we can design many things very nice theoretically, but if the farmers don't adopt them, there's no point for the production. And how we can create habitats, diverse habitats for flora and fauna, and of course, how to move this knowledge and information to the farmers and the stockholders. The stockholders are important because areas, I don't know so much high-end, but for example, in Crete now, agriculture is secondary for the income of the region compared to tourism. So we have also the stakeholders. The tourism industry is quite important. So how we can promote this diversity to the tourism and link it to the tourism and the industry and the other sectors of economy. So I'm not going to talk about this, but just is important, the connectivity, how to reconnect fragmented habitats and to support Natura 2000 areas. And this is important because we see now in Crete, Natura 2000 areas, the neighboring I saw in the map, are more degraded than the olive orchards. We have documented this. And this simply because of overgrazing, and this is due to wrong application of policies, national policies. So we have also this. So what is our approach is green infrastructure consists of two elements. We have elements that we construct in order to manage the native flora, the ground cover already was mentioned, between or around the olive orchard. And this can be done with hedgerows and strips along or inside. And this is important for us because actually olive orchards in Crete is like not in Jaén or other areas. The average size is 0.25 hectares, so very small. So in order to have an impact is how to increase diversity in such a small field and to conserve it. So that's one thing, the things we construct, and the other is the application of sustainable agroecological, what we call agroecological farming practices, because there's no point if we design something or we make some interventions, we put some trees, some flowers and so on, or so ground cover, and then there's no, the farmer just destroy or does not support with appropriate farming practices. So the practices we combined with, we believe that this should be combined. So the canopy management, it was already mentioned before about recycling of pruning residues, what we do with the ground cover, the tillage, it was extensively discussed before about the effects of intensive tillage and soil management, the nutrient fertilizer management, because we have, for example, the most predominant flora species is it's anthropogenic, they're anthropogenic, and they occupy the whole ground cover of the orchards, and what we do with irrigation water and so on if we want to promote ecosystem services. So actually, this technical information, in order to pass it to the farmers and also importantly to the farm advisors, in the framework of another Horizon project, Climate Fruit, we create some technical article that is already available, translated. You can download it, it's free, for the main EU Mediterranean languages. So Spanish, French, Italian, Portuguese, even Croatian, and the Greek. So you see on the right side the information, this article, who explains in detail how to be done, how to do green infrastructure. So just to give you a glimpse, it's the same. We recommend to the farmers and to have at least some hedgerows with target species. We'll see them after. We have specific selected target species that are mostly aromatic and so on that relate to the fauna. And pollinators, beneficials, predators, parasites that prey the olive fly, which is the main pest for the olive tree, because it's important for the farmer again, okay, the aesthetics are important, the biodiversity conservation is important, but the farmer wants an income. He wants to have an income. So, and then about the components, the three main components is the stone refuges that especially for the reptiles and the snails, the water ponds that are important, and it was already shown before, but when you have a small field, how you do a water pond. So we try to find technical, simple solutions for the farmers, because water ponds are very important for the avifauna. The birds need water in the long dry summer. So, and also all the other mammals and so on. And the brush piles, which is difficult because there's always the risk of fire. So the farmers are not in favor of brush pile. And then about the farming methods, ground cover is very important. It's already discussed. And I was surprised actually driving miles and miles yesterday and to see just bare land. So the practice should be mowing and let dry, and this also reduces erosion, and already was presented earlier that you don't need just to plow, which is the traditional method. And another way that's often supported and actually advised is to have some things to sow a cover crop. We have done it with a mixture actually of gramine and fabaceae, different species, but this has a cost, and because of climate variability, it's not anymore so efficient. So it's better to have the native vegetation managed. But again, it depends the state of the orchard. If the orchard starts from minimum diversity because herbicides were applied for 20 years, then you need to reintroduce species, and this takes time. It's always the question of time. And also how to manage instead of plowing. Of course, it can be incorporated, but it just can mowed. And the pruning residues the same. So the pruning residues we have estimated, at least for our variety Koroneki, which is the main one in Greece and in the area, with an average pruning, one tree gives 60 kilograms of pruning residues. So if you burn them, which was the traditional way, then you lose all the nutrients, and you got carbon emissions, and we talk about carbon farming. So we recommend just to chop and incorporate. In the past, there was a notion when I was student also at university, that if you leave the pruning residues, then diseases will spread. But with the conditions, in our conditions, there's not such risk. We have tried them. And then it's the agroecosystem services and that all these diversity offers. And this relates to predators, parasites, and there's so much diversity, and we have done quite a lot of survey and monitoring. From our team, there are 3 posters outside, if you like to see, from colleagues about ant fauna, about flora, diversity, the flora, diversity in the olive orchards, and about parasites for the olive fly. So that's it. And the other is about nitrogen. Nitrogen is very important for every crop and for the olives. So we have documented and we have found that there are many nitrogen fixing species, many fabaceae in the olive orchards, and they are native. Actually, in the past, with a former PhD student, we have done an experiment, which was a bit stupid, but okay, that let's, instead of promote the native ones, let's sow a cover crop with a mixture, actually with veg, and let's inoculate with an inoculum of rhizobium. The result was, it has been published, there's no effect. Because these species, actually the native ones, are really adapted to the olive agroecosystem, the local one, and they have the right combination with rhizobium. So they are very effective. And of course, when we talk about ecosystem services or agroecosystem services, in many areas, also in Crete, we have a big vegetative edible flora. So the people are harvesting like Kabourakis or Amaranthus or Dafcus carotas, cutting this. They are foraging in the olive trees. This was traditional. Of course, it has changed because of the new lifestyle, but this is a service that exists, and it's interesting also how to conserve it and how to maintain it, because we have such a rich edible flora species in olive orchards. So there's also the conservation of the diversity overall, not just for the olive trees. And in olive orchards, we have found, at least in our area, a valuable species, Leodicea leodopetalum, which was just because of intensity disappearing. Of course, maybe somebody to say that there's no why to have it. It's genetic diversity. There is there, and it's easy to conserve it. Another aspect of the snail species, actually a colleague and Vasilis was involved, Georgopoulou did a survey of the snail species in olive orchards. In Crete we eat snails, like the French. I don't know in Jaén if you eat snails, but we consume. And it's important that the olive orchards, like the flora and fauna species, we have quite a lot of snails, and some of them, they are endemic to Crete, and some of them endemic to Aegean. So an olive orchard, like Pedro told before, it's actually like a conservation for the diversity, not only for the flora, but for the fauna also. So that's, and then if we pass to the olive fly, which is, because the farmer, the first thing that asks is, okay, diversity is nice, I like the aesthetics, but what about the olive fly? If I have many species, then probably the olive fruit fly will eat my crop. So there are all these predators and parasites, and these are linked to the flora. So we know that the Thymelaea hirsuta or the Trichia graveolens, they host parasitoids of the olive fruit fly. So that's another ecosystem, agroecosystem function. And so there's a link of flora and family. If you don't have the one, you don't have the other. Of course, the difference is the avifauna. The birds are flying, and they will find their source of food. But if we talk about parasitoids and predators, they are local. Because of, in terms of using energy and so on, they will not fly far away. They risk to be eaten, to take lead sibble. So we do, of course, a lot of monitoring also to scientifically value what we do, the derivations. And there's, for example, I'm not going to detail because of the time limitations, but we see that the organic olive orchards, we use them because they apply mostly the agroecological practices that are favorable for diversity. They have higher parasitism rates, and the fruit damage, olive fruit damage, the infestation is similar. So we see that by increasing diversity, this has an effect to the main pest in the olive orchard agroecosystem. Now there's, of course, it's nice to talk as agronomist or ecologist or agroecologist about diversity, but then the farmer is the income. And of course, even these very simple techniques we propose, they have a high cost. They have an economic cost. If you want to apply it, this costs money. And the benefits are mostly long-term, and the farmer, the olive grower, does not pay for this. He will not be paid if he has a rich edible flora. He will not pay if he has many snails. So then it's important the farmer engagement. For this reason, actually, we develop a decision support tool for farmers and for farm advisors. This is a digital tool. It's free to use if you can link it. And so actually what the farmer does, put some, sorry. I have to go back. So enters data, a free map, enters his olive orchard, and then put his practices. And actually there are pictures, and he select if he has some species of flora or fauna. So himself actually introducing the data of his field. And then he got a ranking. So for example, that your olive orchard has been rated with 99%, but this is okay, and so what? So then we give in detail what can be improved. So what, in terms of water sources, you can have small ponds, and then you will increase your diversity and, or add some stone refugia on wooden refugia, some to make improvements for the green infrastructure. That's the first thing. But then again, the farmers say this is a costly thing. So what we have done, this decision tool is digital, and there's a lot of work behind, but it's quite simple to use. And then we validate and give him also the cost. So if you add this and to increase, you will have this cost. So because for the farmer, it's economics also. He has to live from his olive orchard. And so there's the cost estimation, and then we can, actually the next step is to link this with policies about eco schemes, because farmers are paid with eco schemes. So we can tell him, okay, this cost can be covered by this eco scheme of common agriculture policy. So that's a tool that is, can be used by the farmers and the farm advisors, and we find that they like it. And actually, we have produced a policy document for landscape green infrastructure development, because the reason is that it was a deliverer for our project, but except this, we talk about soil health, and there's the EU policy, but for biodiversity, there's the Green Deal, the biodiversity, we talk, but then there's no concrete policy and economic support for the farmers. So the olive growers, so we have produced this policy document in order to give concrete to the policy makers how to promote green infrastructure development and biodiversity in non-protected farming areas. Because biodiversity and diversity in EU at the moment is for natural areas, which is fine, but a large part of landscape is agriculture. So we have done this work. We wish actually to establish a network and to collaborate. There's also an information video that for farm advisors. And so concluding, thank you very much for your attention. I would like to thank the founding agencies, the LIFE Plus program, the Horizon, and the Green Fund in Greece, and the olive farmers and all the team who work with us and does this job. Thank you. Do you have any questions?
All right. Thank you, Manolis. I think these are the very first steps of escalating, development of ecology in agriculture and olive farming. So it's maybe a good opportunity to